We'll be talking today about aromaticity and the reactions of benzene and substituted benzenes in this chapter. I have a slide here, and I'll come back to this slide in a little bit, but this is a summary of what you need to know for the quiz and the exam. And actually, when I come back to it, it may be a little bit different, because what I do is, as I go through the lecture, I modify some of these slides. So let's talk a little bit about benzene. Benzene is an aromatic compound that looks like it has alternating double and single bonds. We talked about resonance in this structure. Kekule's dream is what that was called, where these bonds, redraw that a little bit, bonds will rotate to adjacent spaces, and so you end up getting this as a resonance structure. Now, there's a term, we call this resonance energy, and it sounds like it's something that's a positive value. It actually is a negative value, but we usually talk about it in terms of a positive value. Resonance energy is the stabilization a compound gets, or a compound receives, as a result of resonance. And the way we learned about this in the previous chapter is delocalization. So that's what resonance energy is. So we're going to look at benzene's resonance energy, that is how much stability it gets by having resonance of those three bonds. So we look at hydrogenation reactions. We've looked at these before in alkenes. And one of the things that if you go back, you'll see for a single bond, you get about 28.6 kcals of energy released per mole. That is, you have cyclohexene, right? This is the ene, the alkene, and this is cyclohexane. That's the alkane. And when you do the addition of hydrogen, usually using some catalyst, like palladium catalyst, what you end up getting is some energy out of that. And in this case, it's 28.6 kcals per mole. Now, since this energy is released as a result of the formation of the alkane, right, this molecule is lower in energy. And that's true anytime you release energy. This is an exothermic reaction, which means energy is released in the process of the reaction. The reason for the lower energy is because in essence, you replace this sigma bond, sigma plus a pi bond, you replace it with two sigma bonds. So what ends up happening here is you end up having extra sigma bonds. And if you remember, sigma bonds are stronger or more stable than pi bonds. So now, in order to understand the significance of being aromatic, what we usually do is we look at the stability of benzene in comparison to its predicted stability. So we know from our previous slide and from hydrogenation of benzene that you get about 28.6 kcals per mole of energy released. So now what we can do is we can look what happens when we have one double bond, or two double bonds, or three double bonds, like in the case of benzene. Now, there's a difference between structures when they're either conjugated or isolated, and this middle structure could have been drawn conjugated the way it was or isolated, but the difference in energy between conjugated and isolated is relatively small in comparison to the difference in energies when we're looking at 2 double bonds versus 3 double bonds. So what we can do is we can predict how much energy would be contained within 3 pi bonds simply by taking 3 times the value of 1 pi bond. So I have 1 pi bond, I have 2 pi bonds, and 3 pi bonds. And then we actually look at benzene's energy. Benzene is 49.8 kcals per mole. Now, this ends up being a resonance energy, which we'll do the calculation on this slide, where I've summarized those calculations, right? Benzene is actually a little bit more stable than having 2 pi bonds. So there's about a 36 kcal energy difference between benzene and what we predict benzene's stability to be. So this extra stability, this gain in stability, this is the significance of aromatic compounds. And aromatic compounds all have large resonance stabilization. Now, large, again, in comparison to our predicted energy. Stabilization. I think it's useful and a little bit informative to sort of understand where this term aromatic comes from. Originally, the idea of aromatic came from the idea of things that smelled good. So the scent of cinnamon or vanilla, these are things that were originally associated with the term aromatic. Later, it was found out that those compounds contain within their structure benzene rings. So now when we talk about aromatic compounds, what we're really referring to is things that have this type of conjugation pattern within them, similar to that of benzene. Methamphetamine, for example, has a benzene ring in it, but so does adrenaline, which is a naturally occurring aromatic compound, and aspirin, which we synthesized from salicylic acid, originally isolated from willow bark. Now, how do we tell if something is aromatic? And there are a couple of simple rules that you need to be able to apply. The first one is the molecule has to be flat. So what does that actually mean, right? When we say a molecule is flat, let's say we're in reference to this molecule. What do we mean by this molecule is flat? Well, this carbon is sp2 hybridized, and this carbon is sp2 hybridized. So this molecule has, in sp2 hybridization, these bond angles are 120 degrees, and that part of the molecule lies within a plane. We also know that if both of these are sp2 hybridized, all the components, not just this part, these parts lie in a plane, but all the components of the molecule, including this hydrogen, which I didn't draw, would be within one plane. So for a molecule to be aromatic, simply put, the molecule needs to be flat.
Continuously conjugated is our next criteria. Now, you can have systems that are conjugated. For example, this is a conjugated system, as is this. So you can have lots of different conjugated systems, but to be continuously conjugated, it actually means that the structure has to be in the shape of a ring. So things like benzene, right, are continuously conjugated because it's a closed loop. Every atom, just like here, each of the atoms that I'm putting this little dot on, these are all sp2 hybridized. Every atom in this part of the structure is sp2 hybridized. Benzene is continuously conjugated. Hexatriene, the molecule that's shown here, that's conjugated, but it's not continuously conjugated in the sense that it's a ring. So the molecule has to be flat. It has to be in the shape of a ring, and every atom within the ring has to be sp2 hybridized. Now the last criteria that you need to satisfy is that you need to have an odd number of electron pairs. So, for example, in benzene, we have 1, 2, 3 pairs of electrons. So there's 6 electrons. That's not the odd part. It's that there are 3 pairs. And it turns out having 3 pairs or 5 pairs or even 1 pair are the criteria. If it meets those criteria, then it's going to be aromatic. So you have to meet the criteria that it's flat and continuously conjugated, and then you have to meet the criteria that has an odd number of electron pairs. These together are actually known as Huckel's rule, named after the guy who originally proposed them. We really only touch on the surface of why the odd number of electron pairs makes it aromatic. We just use the rule, really. So let's look at this, just to show you how to judge whether or not a compound is aromatic. So benzene is it flat? And the answer is yes. Continuously conjugated? The answer is yes. Odd number of electron pairs? The answer is yes. This compound is aromatic. Now, cyclobutadiene, flat? It turns out it's pretty flat. There are reasons why it's not completely flat, but it is pretty flat. Continuously conjugated? Yes. Odd number of electron pairs? No. It has 2 pairs of electron. And so this compound is not aromatic. Now, cycloheptatrienyl, I had to get that straight. Is it flat? It turns out no, it's not. And is it continuously conjugated? The answer is no. If you remember a discussion about cyclooctatetraene from before, there's a bend in the molecule which doesn't allow the p orbitals to align, and so it's not continuously conjugated. And does it have an odd number of electron pairs? And the answer is also no, because it has 4 pairs, which is an even number, and that means it's not aromatic. Now, what about a molecule that looks like this? So we have, is it flat? And it turns out it is flat because every atom in this structure is sp2 hybridized, so it's flat. It has continuous conjugation. And if the pi electrons, we have 1, 2, 3, 4, 5. And so it has 5 pairs. And so this molecule turns out to be aromatic. Now it turns out this is known as the bicyclic compound. These rules don't always apply completely to bicyclic or multicyclic compounds, but for some of the smaller ones, it's fairly easy to apply Huckel's rules and determine whether or not they're aromatic.
So it turns out ions can also be aromatic. So I want to make clear first that even though aromatic ions are relatively more stable than ions without aromaticity, they're actually all unstable in terms of relative to the molecule that the ion came from. In general, ions are unstable. Cations and anions are unstable. So let's apply the rules we learned for determining aromaticity to the cation and anion of cyclopentadiene. So remember, cyclopentadiene has 2 hydrogens attached to it here at this point. So that makes that carbon sp3 hybridized. Now, if I remove the hydrogen and the electrons in the bond, I can get the cyclopentadienyl cation that's shown on the left. So I have a hydrogen that's actually sticking here, and then I have a positive charge on that carbon. Now, because I have only 3 groups attached at that point, there, and 3 groups here, 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 and here, this molecule is continuously sp2 hybridized or is continuously conjugated around the ring. So it actually meets the first two criteria that we talked about. It needs to be flat, needs to be continuously conjugated. It doesn't, however, meet the third criteria that we put up, and that is it only has 2 electron pairs. There's one here and one here, that pi bond. So 1, 2, even number of electron pairs. And so the result is it's not aromatic. Now, if I just take the hydrogen off and leave the pair of electrons, I'll have something that looks like that, a lone pair. Again, I'm not drawing the hydrogen that's there. I guess I could draw it like this. All right. And a lone pair like this. That's my lone pair. So, this, again, is continuously conjugated. You can draw resonance structures, the allylic electron pair pattern that looks like this. And you can see in many of the resonance structures, this particular carbon only has 3 groups on it, so it's sp2 hybridized. So the result is it's flat, it's continuously conjugated, and also because of the lone pair of electrons that I have in the structure, I actually have 1, 2, 3 pairs of electrons involved in the resonance structure. It's an odd number. This structure is aromatic. Let's look at one other case. And that's the cycloheptatrienyl trienol cation. Okay, so the cycloheptatrienyl cation is also aromatic. You'll notice that it has 3 pi bonds in it, 1, 2, 3. It is continuously conjugated all the way around, and you can draw resonance structures for this one. There isn't a pair of electrons here, but there is a p orbital because this atom is sp2 hybridized. So I have 3 electron pairs. I have flat and continuously conjugated. So this guy is aromatic. On the other hand, if we look at the anion is not aromatic. It also has the 3 pi bonds, 1, 2, and 3, but it has a pair of electrons here, which makes a 4th pair. And as a result, it's not aromatic. Now, an interesting thing about these kinds of molecules that are a little bit larger, if you can make them aromatic, they will actually flatten out more in order to maintain the aromaticity. And if they can't, they tend to bend more to break the conjugation. Anyways, this one on the left is aromatic. This one is not aromatic. Here are a couple of other examples of polycyclic aromatic rings. Naphthalene is the one that I showed you earlier. And when you look at the electrostatic potential map that's shown on the left over here, what you notice is that the electrons are distributed continuously around the entire molecule. On the other hand, when you look at azulene, you'll notice that there's that the electrons are distributed in general around the aromatic structure, but a bit more electron density is actually located in this part of the molecule over here. The unusual electrostatic potential map for azulene, unlike naphthalene, which distributes the electron density over the entire molecule, you'll notice in azulene's case, most of the electron density is situated over the left-hand side of the molecule. That's the red that's shown over here. That actually comes from this particular resonance structure, or can be thought of as originating from this original resonance structure when it looks like this. So I'll draw that very quickly here. Let's see, I've got pi bond, pi bond. Sorry, my computer suddenly slowed down. There, like that. I have a negative charge here and a positive charge here. So what that means is this positive charge by resonance can delocalize around the molecule on this side, and this is the cycloheptatrienyl structure that we saw before. So it in itself, that substructure in the molecule, is also aromatic. The result is, because there's an aromatic ring, within the ring, electron density is high on this end of the molecule because the electrons want to delocalize onto this side. And this side ends up being relatively more positive because the positive charge that's created by that distributes by resonance in this fashion. It turns out rings with atoms other than carbon in them can also be aromatic. We call these heterocycles. That is, they have an atom that's different than carbon in the ring. The tricky part in heterocycles is determining which electrons can contribute to resonance or delocalization, and which ones, and then from that, which ones we count in terms of determining whether or not it has an odd number of electron pairs. So we have pyridine. This is an aromatic heterocycle. It has 1, 2, 3 pairs of electrons. It's continuously conjugated. It has the same structure as benzene. But then the question is, does the lone pair participate? And to understand that question, really, or to get to the understanding of that question, we really have to consider what orbital is that lone pair in. In order for it to be participate in resonance, it has to be in a p orbital. Now, when you look at this orbital on the nitrogen, right, it is sp2 hybridized. If you were to draw pyridine on its side, something like this, what that means is the lone pairs of electrons are sticking out to the side of the nitrogen rather than into the p orbitals. These would be the p orbitals that are involved in the aromaticity or delocalization. So this particular pair of electrons can't participate in resonance because they're not in this p orbital. Now, if you look at Pyrrol is another interesting case. Pyrrol is aromatic, and so what that means is you have 1, 2, which would be an even number, 3 electron pairs. So how do you know that that pair of electrons is involved in delocalization? Because it looks like that nitrogen is sp3 hybridized because it has 4 groups on it. The way that you resolve the issue is what you do is you try to draw resonance structures from it. So if you're going to draw resonance structures and you can push this pair here, this pair would have to go here. That would give you a structure that looks something like this. Again, this is that same allylic lone pair pattern that we looked at before. But by being able to draw that resonance structure, what we find is that that pair of electrons is involved in delocalization, because this nitrogen in many of the resonance structures is actually sp2 hybridized. In thiophene, or sorry, in furan and thiophene both, these are also aromatic, but you'll notice there's 2 electron pairs, and only one of these can contribute at a time. So for furan, it'll create a structure that looks like this. Thiophene will be very much the same. Negative charge like that. And you'll notice, it's a very unusual drawing, sorry, let's fix that up just a little bit, something like that. You'll notice that this structure and this structure are exactly the same. And if you go back just a couple of pages, a couple of slides, they're the same as the cycloheptatrienyl anion, except for the cycloheptatrienyl anion is not a heterocycle, it just has a carbon in that same position. So the delocalization, if you can delocalize the electrons, I should say, into the ring, then they can participate in resonance, and then they count as one of the electron pairs in determining whether or not it's aromatic. Now you'll notice when I do that, this pair of electrons is in an sp2 hybridized orbital, and the delocalization is occurring only with one of the 2 pairs of electrons. So you have 2 pairs of electrons here, and you have 2 pairs of electrons here. Only one of those 2 can contribute, and when they do, it creates a situation where the heterocycle is aromatic. In general, what I tell people to do is if a lone pair can contribute to resonance stabilization, let's just say resonance, right, if you want to, or stabilization, it will. And then further, if that counts as the odd pair of electrons in your electron pair count, then that can make it aromatic as well. So a heterocycle is a ring that contains an atom other than carbon. So nitrogen, oxygen, sulfur are very common in organic chemistry to be the atom that is the hetero atom. Heterocyclic aromatic ring simply means it's an aromatic ring that has an atom other than carbon. So pyridine is a very common example. There's a nitrogen in the ring that makes it a heterocycle. One of the things that you need to be able to do is be able to determine whether or not heterocyclic rings are aromatic. And it can be confusing because if you look at the pyridine looks very much like benzene, and you might just naturally assume that it's aromatic, which it is. But it also has this pair of electrons in here, and the question is, why doesn't that make it an even number? And pretty simply put, it comes down to whether or not that pair of electrons can contribute. So if a lone pair can contribute to stabilization, that is resonance stabilization, it will. Now, the main criteria for that is that whether or not that the contribution of the pair of electrons on the atom can be delocalized in the ring. Okay, so when I say it will, it means participates in resonance. Just abbreviate there. Now, Pyrrol, Furan, thiophene, these are all examples of heterocycles that are aromatic. And what I thought I would do is go through a couple of these examples, like one by one, in some details to help you understand. So here's pyridine as our example. Now, how do we know whether or not a pair of electrons participates in resonance? A simple explanation goes, in essence, you really can only contribute one pair of electrons per atom. And in nitrogen, this is really sort of like the second electron pair contributed to the ring. But how do we understand that, and how do we know that that's the case? So for a pair of electrons to be involved in resonance, it has to be involved in the delocalization. And the delocalization occurs in the p orbitals. That means the electrons have to be in the p orbitals or delocalized in the pi bonds of the molecule. So when we think about nitrogen being double bonded, it's got a double bond here, for example, and then there's a double bond here, or a double bond here, like this, for this structure based on the structure down below. Right? These orbitals that are p orbitals are perpendicular to the molecule, and the rest of the molecule is flat. It's all in the plane of the molecule. This pair of electrons is also in that plane because it's bonded to an sp2 hybridized orbital. So we can only have electron pairs per atom. We can really only have one contributing. And the reason the only one can contribute is because the second pair is in the plane of the molecule. And because it's in the plane of the molecule, it can't get into the p orbitals that are perpendicular to the molecule and delocalize. So for pyridine, it's pretty simple. This portion of the molecule that looks like benzene, these electrons that are in the ring, part of them are contributed to the nitrogen. This second pair can't contribute because it's in the plane of the molecule. Now, if you look at the electrostatic potential map for pyridine, what you see is something that's interesting, in that you see this bright red spot that's down here. Now, part of that is because nitrogen is more highly electronegative than carbon, but the reason for the big red spot is primarily due to the idea that there's a lone pair that's localized on that nitrogen, whereas the pi bond is delocalized. Now, the same thing, we could look at the same thing for Pyrrol and Furan. In Pyrrol, we have a pair of electrons on the nitrogen, and we have also on the nitrogen we have a single bonded hydrogen. This pair of electrons can contribute to the delocalization by resonance, and we can draw resonance structures for Pyrrol. The same thing is true for Furan. We can contribute one electron pair. And when you do that, and I'll show you this on a later slide, the other pair of electrons, and it doesn't really matter which pair you start with, but the other pair of electrons will be in the plane of the ring. So let's draw the resonance structures for Pyrrol. I drew the skeletons up to make this go a little faster, but one involves the pushing of the electrons from the nitrogen, and then it's an allylic pattern again, so you'll end up with a pair of electrons here, a double bond there, and a double bond like that, and there's a negative charge on here and a positive charge here. So what I'd like you to do is go ahead and draw the remaining resonance structures until you get back to the original structure. So go ahead and pause the video, and then when you resume, I'll have all the structures drawn on the board for you. So here are the arrows for the delocalizations of the electrons, and you'll notice that this pair of electrons on the nitrogen, right, is pushed into the ring in the allylic pattern, so you get a lone pair here. We do a second push here. This pair of electrons ends as a lone pair here, and then we push it again, and we get down to this structure. We'll push it one more time, and we'll end with this structure, and then when we push this pair back into the ring, this pair goes back to the nitrogen, and I'm back to the original structure. Okay? So there's 5 resonance structures, basically one for every atom in the ring. Now, and that's generally true for rings with odd numbers of atoms in it. It seems to be. Anyways, what you'll notice in the electrostatic potential map is there's a positive charge on this end, and then the rest of the molecule, the other end of the molecule, is red. And the red comes from the idea that you're delocalizing this negative charge that's over here all the way around the molecule. And so every atom, every carbon atom at some point gets touched by this negative charge. And then the nitrogen for the rest of the time is positively charged, so it shows up as a positive charge in the electrostatic potential map. When you're looking at this, any of the resonance structures other than the initial one, this atom is sp2 hybridized. This hydrogen is in the plane of the molecule, and even though it looks like the nitrogen might be sp3 hybridized, the resonance structures indicate that for the predominance of the time, in terms of resonance, it's delocalized as sp2. Now, Furan is a similar case. So what I'd like to do is to have you draw the resonance structures, and just so we're all on the same page, we're going to start going in this direction, and we're going to go to here. So I'm going to pause the video, and then I'll draw all the structures. I'm going to go in this order, okay, like that. And just so you know, right, these are supposed to be resonance arrows, but I'll just put arrows in one direction so that we can all keep going in the same direction when you draw your resonance structures. So you might want to, again, stop the video and check to make sure you drew your resonance structures correctly. But when you continue on, what you'll notice is that in the drawing of the resonance structures, the pair of electrons are delocalized into the ring. I have a negative charge at all the positions around the ring. I have a formal charge that's positive on the oxygen, but I also have the lone pair that's down here. Okay, so when we look at the structure, the electrostatic potential map for Furan, what we see is a red charge here. This is the lone pair and on the oxygen. And also in part due to its sort of more electronegative nature than nitrogen. And then we have a delocalization of negative charge around the ring. These blue portions that are here, these are the hydrogens that aren't drawn on the structure. So, but again, you'll notice too that once I've created this resonance structure where the one pair of electrons is contributed to the ring, this pair of electrons is now in an sp2 hybridized orbital. And as a result, because it's in the plane of the ring, it also can't contribute to resonance. And so we only contribute one of the two electron pairs on the oxygen. In general, atoms can only contribute one pair of electrons to the aromaticity or resonance of a structure. So the naming of benzene is relatively easy, but it's confused by the presence of a lot of common names. And oftentimes the common names are what people really know substituted benzene as. Typically, when you name benzene, you just say the name of the substituent and then you add the word benzene to it, and that gives us the name. So this would be, for example, the ones I've circled, ethylbenzene, because that's an ethyl alkyl group, or nitrobenzene. It was the same as naming alkanes. You name the parent, so benzene is the parent. And then there's a substituent on it. The problem is, and I've highlighted this on the next slide, anisole is a very common name for people in organic chemistry. It's what we call actually methoxybenzene. Then MEO, that's a CH3O group. This is known as a methoxy. So systematically, we would call that methoxybenzene. But I want you to know these common names that I've highlighted for you, because oftentimes when you do the nomenclatures of benzene, it's acceptable to use these common names and treat them like they're part of the systematic name. So, for example, toluene is actually methylbenzene, this guy here. That would be its systematic name. And so if you have a substituted benzene that looks like toluene, you could name it either as methylbenzene, which is actually the correct systematic name, or you could actually call it toluene. Either one is acceptable. But I do want you to know both. Now, it turns out phenol, for example, is the parent name. The hydroxybenzene, you might call it, but the phenol is generally the systematic parent name. But all the others that I've listed here are common names. So there's a lot of different common names. I would like you to memorize these particular ones. There's a lot more, but these are the ones I'd like you to memorize. Now, let's talk about the reactions of benzene. So, when we looked at electrophilic reactions in the past, what we actually looked at was electrophilic addition reactions. This would be an example of an electrophilic addition reaction. The pi bond is broken by the electrophile, and then the nucleophile is popped off. And then what that will create as an intermediate is an electrophile attached to a cyclohexane in this case, and a carbocation. I drew the opposite of what I have drawn on the product. I have the electrophile here. The carbocation would be there, and then the nucleophile would come in and it would attach itself to that point. So this is the standard electrophilic addition reaction. It starts with the electrophile, and it ends up as an addition because you add both the electrophile and a nucleophile. It doesn't necessarily have to be the nucleophile that's created by the electrophile, but an electrophile and a nucleophile to the ring. When we're looking at aromatic compounds, what we actually get is something slightly different. What we get is what's called an electrophilic aromatic substitution reaction. Now, it's called the substitution reaction because in the process of the reaction, you take a hydrogen that is attached to the ring and you replace it with the original electrophile. And in the process, we don't do an addition; we do a substitution. So that means all we've done is substituted. There's still the pi bond present from the original ring. So, in a sense, this turns out to be what's called an addition-elimination reaction, and I'll show you what that means in a second. But why doesn't the elimination take place? Okay, so let's look at this mechanism. So for the addition reaction, the product will look like this. Now, I'm not drawing on the structure down below because I'm going to draw the mechanism and look at the intermediate and talk about the route to get to the addition product. But the product is going to look like this. I'll have an electrophile attached, a nucleophile attached, and the pi bond is now gone. So in the mechanism, the pi bond attacks the electrophile. I'll draw the hydrogen that's here too, even though I didn't draw it in the original one, but it helps to make sense of the structure. When that pi bond breaks, the electrophile is attached to this point. It could be attached to the other one, but I attached to the top one. And then what else you have is that you have a positive charge at this point here. Now, at the same time, you have a nucleophile in solution, or in the reaction. And the nucleophile with its negative charge comes along and attacks at this point and goes to form the product, and this would be the addition product. Now, this intermediate, actually, there's a couple of choices as to what happens with it. The addition product is what I've drawn, but it can also go through and form an elimination product. And so I'll show the elimination step on the next slide. So this is how the elimination might take place. You have your nucleophile, and instead of acting as a nucleophile, and again, this is the same structure that I drew on the previous page as the intermediate. The nucleophile can be acting as a base. And because it's acting as a base, it's going to grab this hydrogen and then kick the electrons back into the ring. And then what you've done is by removing the hydrogen, you've ended up substituting in net, you've ended up substituting the electrophile for the hydrogen that was originally on that ring. And so this would be, overall, this is, even though it takes place from the last portion of the mechanism, which is elimination, this is a substitution product. Now, regardless of which mechanism we follow, it's important to understand that you can break the pi bond because that structure is stabilized by resonance. That is, if you think of if you think of, oh gosh, these pi bonds can be moved, and if you wanted to, you could draw the resonance structures like this. That positive charge gets delocalized throughout the ring. Now, let's talk a little bit about why we go by substitution in general rather than addition. So here's my addition product, and here's my substitution product. And even though these are conjugated, remember, conjugated double bonds do have some stability associated with them. It turns out the substitution product is aromatic. And this is the importance in talking about resonance stabilization in relationship to aromatic compounds. The stabilization gained by an aromatic compound, while it delocalizes its electrons, causes the substitution product to be far less energetic than the addition product. So once you get to this intermediate, which could either undergo an addition or a substitution, it chooses to undergo substitution just because the product is more stable than the addition product. Now, in terms of energy, because it's lower in energy, its activation energy is generally much lower. And we've talked about this in earlier chapters. A lower activation energy means the reaction happens much more quickly as a result. Now, there are a number of different reactions that I want you to memorize, but I don't necessarily want you to memorize all the mechanisms. So there's major reactions you need to memorize, but you don't need to memorize all the mechanisms. What I want you to do is I want you to memorize this general mechanism for the formation of the intermediate and the elimination to form the substitution product. So it's really a two-step mechanism. It's the electrophilic attack followed by the elimination to form the substitution product. Now, there are a number of reactions that you need to memorize in this chapter. Some of them are shown here, but I want to emphasize, while there's a lot of reactions here, you don't need to know a lot of the mechanisms. The mechanism for all of these essentially follows the same route, and so I'm going to draw that for you on the next slide. And I'll give you an example of how you can maybe more easily memorize these different reactions. So the mechanism that you need to memorize is the formation of the substitution product. So, for example, here's a benzene ring. And you can draw hydrogen on it. Now the hydrogen's there. I mean, you know that that's there, but we only draw the one on there because that's the one involved in the substitution portion of the reaction. So first step is you have an electrophile, and oftentimes people will use the Y plus as the electrophile. Then this bond breaks and it attacks the electrophile, and you form a carbocation. So in this particular instance, the first intermediate we draw looks like this. And I'll draw the double bonds that I didn't break back on the ring, and then the other carbon that's left is the carbocation. So to make that clear, I drew the structure exactly the way it was in the bond I attacked. One end has the electrophile, which is Y, and the other end is the carbocation. And there's the hydrogen that's here as well. Okay, so everything's the same except for the broken pi bond and the carbocation and the added electrophile. Now, the second step in the mechanism is a base, and the base is usually some other molecule in the solution, like what was with the electrophile. And that bond is broken between the hydrogen, and then you get back your substitution product. So that's the general mechanism for electrophilic aromatic substitution. So I'll just write EAS mechanism, okay? And give you an example of, and a little bit more information that you also need to be able to do. You need to be able to draw the resonance structures of this. So one of them, for example, would be like this, to show why it's you're able to form it. That is stabilization by delocalization. And then this one comes over to here, and then you have another resonance structure like this. So as part of the mechanism, you also need to be able to show its resonance structures for the intermediate. So you have this intermediate in the middle, it has 3 resonance structures. Okay, so how can you memorize those reactions a little bit more easily? So again, I always recommend that people physically make flashcards. Don't rely necessarily on a program, because part of the memorization is actually writing it out by hand. So you'll have a flashcard, and this is a 3x5 card. So this is side 1. This is side 2. And what you're going to do is you're going to put benzene on one side. And again, the circle is a shortcut notation for the double bonds. It's a little faster to draw. And then for the halogenation reaction, for example, the one that I show here, you notice I said Fe, X3, and X2, and it puts an X on the molecule. So the X is any halogen, really. So let's say it's Fe, Br3, and Br2. So that's what you have on one side. And on the other side, you're going to have this. Oh, sorry. Got all these stray marks on my page. Sometimes you'll see stuff written down at the bottom over here, and typically those are solvents. So you notice side 1 and side 2 kind of look the same, and then over here I'm going to have this. And this is how you can use the card. You look at the product and the reagent, and then you can figure out what the reactant is, or you can look at the reactant and the reagent and figure out what the product is. Another way to do it is on the reverse side of the card, is you don't write this, but you write this. And the reason this kind of card is useful, you have the reactant and product, but you don't have the reagent. So what you do is you look at side 1 and you predict the product, and then when you go back through the cards, you go back through them when they're backwards, and you try to write out the reagents on a piece of paper that give you this product from this reactant. Okay, so of the 2 ways that people tend to make these cards, I kind of think this second way is probably the most efficient way. That is, you have reactant and product and no reagent, so you know that you have to supply that. Or you have the reactant and the reagent, and then you have to supply the product. There are other permutations of that, but I kind of think that if you can do that, you can do most of it. Okay. So the next thing that I want to do is show you how some of these different electrophiles get made. So in halogenation, the primary electrophile is essentially a bromine as a cation. In other words, it's a Br+, and it's very electrophilic. And the way that it's actually produced is it's actually not produced as the cation, but iron acts as a Lewis acid, and what happens is this bromine will attach to the iron. That would be this bromine. I've kind of drawn it in reverse here, and that creates a partial negative charge on this side, and a very strong partial positive charge on this side. So essentially what that does is it weakens this bond and creates a situation where the bromine can be easily grabbed by the benzene ring. But for practical purposes, the bromine, the electrophile, looks something like that. Now this is actually true for all the halogens, so you could substitute with bromine or iodine or with chlorine or fluorine. Any of the halogens will act in this fashion. There's some details that are not important for us about how they're different, but for the most part they're all done the same way. Now, in nitration reactions, a nitration reaction will look something like this. Oh gosh. In nitration, the nitration will be benzene plus, and then an arrow, and then typically it's HNO3, so it's nitric acid and H2SO4, just a mixture concentrated, and then it's heated, and then what you'll get is you'll get a nitro group on it like this. Okay, so that's the nitration reaction. I showed the halogenation reaction as an example earlier. Nitration would be like this, and again, you would make flashcards with reactant and product on one side and reactant and reagent on the other side so that you could use that to help you memorize these reactions. Now, again, I don't necessarily want you to know how all the electrophiles are made or what the electrophiles are, but I do want you to know how the nitronium ion gets made. Nitronium is this guy here. This is actually the electrophile in the electrophilic addition of a nitration of benzene. So the way it works is that nitric acid and sulfuric acid, when they're mixed together, the nitric acid will protonate, steal a pair of a proton off of off a sulfuric acid molecule, and you form this charged species. And if you look at this, right, this is actually water on the structure. So what ends up happening in the next step is that this bond breaks and goes back to the oxygen, and this is the water molecule that we see over here, and the other product is the electrophile for the electrophilic addition of a nitro group to a benzene ring. Okay, so this is one mechanism I actually do want you to memorize, and again, it doesn't hurt to use flashcards, a small set of flashcards to help you do that. So the mechanism looks like this, right? And then when I break the bond, I get a positive charge over here. And then I have this hydrogen sulfate, which is the most likely base in the solution. It attacks that hydrogen and brings those pair of electrons back. And you end up with a nitro group on the ring. So another important reaction is the sulfonation reaction. It's summarized here. Oh, sorry, where'd it go? Oh, here, the sulfonation reaction. And it requires sulfuric acid and heat. So let me write that reaction out for you on this slide. So you have benzene, H2SO4, and heat, and then you ends up with a benzene sulfonic acid. That's the name of that guy. But it's a sulfonic acid is like sulfuric acid, but on one end of it, it has a benzene ring. Or another group. The mechanism for the electrophile, and again, you don't need to memorize this electrophile, but you do need to memorize that reaction. The first part of it is actually this. And what that ends up doing is it ends up protonating this oxygen. And so you'll have a positive charge here. And just like in the nitration reaction, this bond will break and go back. So in addition to the other product, you have water. This is the electrophile. Now, I should point out there's a positive charge here because when that oxygen takes the pair of electrons, what ends up happening is you have to leave a positive charge somewhere, so it ends up being on the sulfur. Now, as the electrophile, and I should also point out there are other ways to draw this mechanism. This is the route I want you to go through, but if you look it up, you'll see that also another possibility for the electrophile that's formed is actually just SO3. This is sulfur trioxide. And you can kind of see how that is if you pull this hydrogen off. That's how you get that. Now, and either can be used as the electrophile in the reaction. So the electrophile reacts with a benzene ring by breaking the pi bond, and that pi bond then attaches to the sulfur and then leaves a positive charge on this carbon. Again, in the mechanism, we have a base that comes in. The base attacks this hydrogen and then kicks the electrons back into the ring. And so it's the same kind of mechanism that we see for both nitration and halogenation. It's attack the pi bond in the ring, and then a base, a molecule with a pair of electrons, comes and pulls the hydrogen off as an elimination. So this is that step, right? The base is probably this hydrogen sulfate, comes in and attacks this hydrogen, that kicks these electrons back, and this is your substitution product, your sulfonic acid. Now, Friedel-Crafts acylation works very much the same way. It's a little bit of a different kind of reaction. In the Friedel-Crafts acylation reaction, we're taking something that looks like this. It's a acyl halide. This is what it's called. This can be X group. Now, typically that's a Cl. And then we react it in it with a benzene ring. So we would write it something like this in terms of R and then C double bond O Cl. And then there's a catalyst for this, and it's aluminum chloride, like that. The product will be this. And so what it does is it substitutes an acyl group. That's an alkyl group or a group with a carbonyl on it, a C double bond O on it, for the hydrogen on the benzene ring. The mechanism is exactly the same. The pi bond is broken, attacks the carbon of the electrophile. The electrophile forms because the aluminum chloride is very good at pulling the chlorines off, and so it pulls the chlorine off and forms this known as an acylium ion, which has a resonance structure that looks like this. It makes the carbon very electrophilic, and as the electrophile is able to break the pi bond, and then we get a subsequent elimination reaction to produce the acyl substituted benzene ring. Now, there's also an alkylation reaction. We don't see that as much, but in an alkylation reaction, you use an alkyl halide like this. And so it's not an acyl halide. And an alkyl halide, when it reacts, will react in very much the same way that an acyl halide will. The aluminum chloride, again, acts to remove the chlorine, or to break or weaken the bond between the alkyl group and the chlorine, and essentially it reacts as a carbocation. Now, the carbocation is a very strong electrophile, and it's able to break the pi bond. There's a subsequent elimination group reaction, and you add the alkyl group onto the ring. Now, the one danger of using alkylation reactions is they typically don't use it for primary alkyl halides because there's a lot of what we call rearrangement. The alkyl group will rearrange when it attaches, and you don't get the group that you're necessarily hoping for to get attached. You don't need to know that particular detail. If you take the Chem 28 series, that's where you'll learn those details of like the rearrangements of the substituents that you add. In naming disubstituted benzene rings, we follow the same protocols that we've used before. You give the first substituent or the parent functional group the lowest possible number on the chain. In this case, it's a ring, so it would be 1. And then we would say 2, 3, 4, 5, 6. And then we use benzene as the parent name. And this one would be 1,2-dichlorobenzene. If you only have 2 substituents, we often use the designators O, M, P for ortho, meta, and para. Ortho is 1, 2, meta is 1, 3, and para is 1, 4. Orientation on the ring rather than using numbers. I've added a few notes to this slide. Whenever you name disubstituted benzene rings, we can either use the numbers to indicate the substitution pattern, or we can use the designators ortho, meta, and para to designate where the substituents are on the ring relative to each other. So this first compound on the left, 1,2-dichlorobenzene, could also be called O-dichlorobenzene, which stands for ortho, which means next to each other on the benzene ring. Meta would be the 1, 3 position, so this is 1 and this is 3. This could have also been numbered 1, I just counted it in the opposite direction. But this could have also been the carbon 1. But meta orientation, meta is sort of thinking about in the middle, could be used as well as 1, 3, or M-dichlorobenzene down here is the abbreviation of meta. Para, you could think of the lines being in parallel with each other. But these are 1, 4, or P-dichlorobenzene. If you want a little explanation of this picture in the corner here, you can look that up. It's in your book. It's kind of funny. Anyways, relative positions you need to get comfortable are often indicated with ortho, meta, and para. Now, toluenes, phenols, anisols, anilines, benzaldehydes, these are generally named as substituted versions of those particular substituted benzenes. So where we learn methylbenzene as toluene, when you're going to name a substituted methylbenzene, we generally name it as a toluene. So this is 4. This is 4. 1, 2, 3, 4. 4-bromotoluene. Or we can use the designator para because they're in that 1, 4 orientation opposite each other. Or 2-nitrophenol. That would be 1 and 2. Can be used as well because this part of the molecule is phenol and then this part of the molecule is toluene. So when we looked at the reactions of benzene rings, all the substituents were hydrogen. But if you react a substituted benzene ring, then the reactivity of the ring generally changes depending on the type of substituents. And we classify the substituents actually 2 different ways. Activating means reacts faster. And deactivating means reacts slower. And what we mean by reacts faster or reacts slower is we mean that it reacts faster than just benzene by itself. So one way, again, we look at reactivity. We say faster or slower. We have activating and deactivating substituents. There's another classification which is also very important. And these are whether or not it's an ortho-para-directing substituent on the ring or a meta-substituent, directing substituent on the ring. So to give you an example, if I take a benzene ring and I put a OH group on it, and I do an electrophilic aromatic substitution reaction with this phenol now, the most likely positions for the substituent, the electrophile to add, the next substituent to add, would be either in the ortho or the para position. And let me draw two different ones, because normally we talk about adding one or the other, although oftentimes you get both adding to the ring. Okay, so you can have it like that or like this. So this is what we mean by ortho-para directing. The major product has substituents when they're added for the second substituent in the ortho and para direction. A meta-directing substituent is actually something like a nitro, and we'll go over these in the next couple of slides. I don't expect you to memorize whether or not different substituents, particular substituents, are activating or deactivating, or whether or not they're ortho, para, or meta directing. But I will provide you a table with that information. But I would like you to know why they're activating, why they're deactivating, and why they're meta or ortho para directing. But a meta director, like a nitro group, when we do an EAS reaction to it, will generally add the second substituent in the meta position, so like this. Okay? So those are the two ways we classify it. Makes it faster, makes it slower, that is activating or deactivating, and whether or not it's a meta director or an ortho-para director. So one of the ideas that I wanted to get across is that when we talked about activating substituents, we're really talking about substituents that increase the electron density in the ring. So those types of substituents generally have two different kinds of features. There's some which are alkyl substituents, and they donate electron density through something I mentioned briefly earlier called hyperconjugation, but it's through the bonds of the atoms associated with the carbons on the alkyl group. But the other kind of very common pattern that we see is that we see that they donate electron density through resonance. So I've drawn here the 5 resonance structures for anisole. Anisole is this methoxybenzene. And you'll notice here that electrons get donated from a lone pair, and that's one of the key features, is that you have a lone pair, into the ring. And then what happens is the lone pair, which we're drawing here as just a line, travels around the ring until eventually it goes back to the oxygen, and you're back to the original structure. The bonds are rotated, so we could do that Kekulé shifting of the bonds around the inside and then get back to the original molecule. So before I say much more about it, what I'd like you to do is I'd like you to pause the video and draw the resonance hybrid that you would imagine this would look like. So here I've drawn the resonance hybrid, and what do you notice about the electron density in the ortho, para, and meta positions? Right? Well, when I draw the hybrid, first of all, I have a negative charge at this carbon in one resonance structure, this carbon, and this carbon. So those are the ortho, right? These are the ortho, and this is the para position. So what an electron donating substituent does primarily is increase electron density in the ortho and para positions. Now, it also has a slight positive charge on the oxygen because of the double bonded oxygen that we created up here. But the important thing to recognize is by putting electron density in the ring, we increase the availability of electrons for an electrophilic reaction. Now, it turns out the fact that these electron densities show up in the ortho-para position means that it's able in its intermediate, and we'll show this later, but in the intermediate, able to stabilize the positive charge that's created there during the addition of the electrophile. So we're looking at The kind of substituents that primarily do electron donating are alkyl and those with lone pairs next to the ring. Now let's look at an electron withdrawing substituent. Now a nitro group is an electron withdrawing substituent, and there are two general features that we see in electron withdrawing or deactivating substituents. And those features are high electronegativity, and pi bonds on the atom attached to the ring. So in this case, in a nitro group, the double bond on the nitrogen, this is the pi bond on the atom attached to the ring. Okay. The nitro group is able to withdraw electrons through resonance. So here are the different resonance contributors for the resonance hybrid, and I'd like you to pause the video and quickly draw that resonance hybrid. So I've drawn the resonance hybrid, and what I want you to do is focus on the positions of the positive charges in the ring. Right? Those are the same positions that we find the positive charges in the resonance contributors, and so we put partial positive charges all around the ring. Now the nitrogen turns out to be an unusual one. It always has a positive charge. So in every single structure it has a positive charge, so I just put a positive charge there. But the oxygens share in the carrying of the negative charge on the nitro group. So I put partial negatives. You'll notice this one's negative here, and then this one's negative here. So what does this positive charge do? Well, remember, in an electrophilic reaction, the electrophile will want to take electrons from the ring. And the result is, by lowering the electron density in the ring, the availability of electrons for the electrophile goes down. Another very important idea is, by putting a positive charge on the ortho and para positions of the ring, when we add the electrophile and create a positive charge in the ring, that's a much more unstable situation and tends to slow down the reaction. So we'll look at some of this in detail in the next couple of slides. Let's look at substituent effects from a different perspective of electrostatic potential maps. Now, we know that anisole, with the oxygen with the two lone pairs on it, is an electron donating group, and that when it does electron donation, that in the hybrid we see partial negative charges in the ortho and para position. There's two ortho positions and one para position. And that's what we're seeing here in the structure when we see the red in the ring. Now, the red that you're seeing right here, those are the lone pairs that are on the oxygen. So those being at strong negative charge show up pretty brightly in the electrostatic potential map. Now compare that to benzene. Benzene is nonpolar. There's no distribution, no uneven distribution of charge in the molecule. And it's given a green color as it is generally neutral around the ring. Now, when we look at nitrobenzene, the nitro group has the two oxygens on it that can then pull electron density out of the ring, both because of their electronegativity, but also because of resonance. So we know in the electrostatic potential map, or in the resonance hybrid for nitrobenzene, is instead of negative charges, we have positive charges distributed in the ortho and para positions. When you look at the ring for nitrobenzene, in comparison to anisole, what you find is, in general, there is much more positive charge in the ring. And remembering that electrophilic reactions require electrons in the ring, we can assume that withdrawing groups generally decrease the reactivity by decreasing the availability of electrons in the ring, and donating groups increase the availability of electrons in the ring. So here are a list of the orthopara directors that you'll get on the test. And I wanted to talk quickly about some of their general characteristics, like I've talked about earlier. Ortho-para directors in general are activating groups. That is, they're able to donate electron density. So you'll notice down here alkyl groups can donate electron density. And then esters are also electron donating. And when you're looking at this diagram, you have to imagine that the benzene ring is attached to that one long bond that's shown to the left. So amines are the most strongly activating. Phenols are the next most strongly activating. But they're also, all of these groups are ortho-para directing. The only one that's deactivating, that's ortho-para directing, is halogens, which, because of their electronegativity, are deactivating. But they're ortho-para directing, as it turns out, because they have a pair of electrons, lone pairs of electrons on their structure. Like that. So let's look at how this happens for ortho-para directors and how the substituents get added in the ortho-para positions. So here's an example of bromination of anisole, or an oxygen-containing benzene ring. So this could be anisole, it could be an ester, it could be a number of different structures. And we have bromine that is used to substitute the hydrogen in either the ortho position or the para position. So these are the two products that you'll get out of the reaction. So at the beginning of the reaction, remember, this essentially acts, Br2 essentially acts as a positively charged bromine, and the bond breaks. And the bromine can either add to this position or this position. So now, if we add the bromine in this position, what happens? Well, if we do that, we get a positive charge on this carbon, and that positive charge can be delocalized around the ring. So that would be the start of the first resonance structure. And what I'd like you to do is go ahead and draw the remaining resonance structures, and then we can talk a little bit about why that might be favorable for substitution in the ortho and the para positions. So here are the resonance structures that are now available because of the oxygen that's on the ring. If you remember when we did the reaction before, there were 3 resonance structures. These were the 3 that we had for the addition of the electrophile. But now with the oxygen on the ring, what happens is there's another resonance structure. So the extra resonance structure adds more stability to the intermediate. So having 4 resonance structures makes the intermediate more stable. So the next question to ask is, ortho-para looks like it has an extra resonance structure if we add it in those positions. What if I added it to the meta position? So if I add it to the meta position, you'll notice the positive charge is on this carbon. And then when I draw the resonance structures, that's the first of the 3 resonance structures that I'd get. Go ahead and pause the video and fill in the remaining structure. So what you should notice is that by adding it in the meta position, I bypass being able to use the pair of electrons on the oxygen to stabilize the charge that was created in the intermediate. So when we're comparing ortho-para addition versus meta addition, then there are fewer resonance structures in the meta position, and the result is that activation or addition isn't as fast as, the intermediate isn't as stable as the intermediate for the ortho-para addition. Now, you should be asking yourself also, going back to this previous slide, what happens when I add it to the para position? Because you notice I only showed what happens when you add it to the ortho position. Now, if I add it to the para position, what will happen is the addition will go like that. And what I'll end up with in my intermediate will look something like this. And you'll notice that that first resonance structure will place the positive charge back onto this carbon where the oxygen is, and the lone pairs on the oxygen can stabilize that resonance structure. And in addition, that also produces a fourth resonance structure which helps to stabilize the intermediate. So the ortho and para addition products both have 4 intermediates, and it's possible to do the additions in those positions more quickly than if you add it into the meta position. This also answers the question of why do halogens ortho-para direct even though they're deactivating? Simply being activating or deactivating isn't enough to create a situation where ortho-para is favored over meta. But because of the lone pairs on the halogen, again, chlorine, bromine, iodine, fluorine, they all have these lone pairs. But because of the lone pair on the halogen, they're able to distribute the charge in the same way that the electrons were distributed when we had other electron donating groups. The result is halogens tend to be ortho-para directing even though they slow the rate of the reaction down. When we look at the electrostatic potential maps of benzene and halobenzenes, in this case chlorobenzene, on a relative scale, you'll notice the benzene in this ring is red, but with the halogen on the ring, it's less red, and there's more electron density towards the chlorine than there is actually in the ring. That difference in electron density causes this ring to be less reactive to electrophiles, and as a consequence, the ring, we say, is deactivated. The halogen, though, because of its ability to donate electrons, like we showed in the previous slide, still acts to direct the next substituent to the ortho and para positions. So now let's take a closer look at meta-directors. Meta-directors all have strong electron withdrawing groups. In fact, one of the strongest electron withdrawing groups is CF3, because the fluorines are so strongly electronegative that this carbon then serves as an electron withdrawing group from the ring. Now, if you look at all the other ones that I've shown here, I have a double bond on this carbon adjacent to the point of attachment to benzene. So the double bond, in this case it's a triple bond. Now the SO2OH, actually, Lewis structure, should look like this. I don't know why the pen didn't work. Like that. Okay. So it has double bonds. And the nitro group, which is the most strongly deactivating group, has a nitrogen with a positive charge on it and a double bond or a pi bond on the nitrogen. These types of substituents all deactivate the ring, and they all are all meta-directing substituents. So there's a lot of information on this slide, but what I wanted to show is the different resonance structures that you got if you put these structures, the intermediate and the resonance structures side by side for both ortho addition or para addition as well as meta addition. And the stuff that I say about para will also apply to addition in the ortho position. So if we look at the meta addition, and we draw the resonance structures, we have the double bonds here. And when we add the bromine in the meta position, what we end up is putting a positive charge here. Now we delocalize that charge like this. And that's where that stops. And so there's 3 resonance structures. You'll notice that the resonance structures bypass putting the positive charge on the carbon where the C double bond O is. Now let's go ahead and look what happens when we do it in the para position. So when you add to the para position, you get a positive charge here. So we're adding it to this position here. The positive charge will show up on that carbon. Now what happens when I do the resonance structures? I go like this, right? And I'll have a positive charge here. And then I can go like that, and then I'll have a positive charge here. Now you notice it also stops at that point. And so I can't draw a fourth resonance structure. And there's also something interesting about these resonance structures. This carbon is electron withdrawing, and it turns out by being electron withdrawing, it's less stable when there's a positive charge here, because with the positive charge, it can't pull the electron density out. So this positive charge makes this particular intermediate less stable. By bypassing this carbon in this position here, then what happens is this set of intermediates is made more stable because the positive charge is never put next to the electron withdrawing group. The result is that adding in the meta position, even though it's slower, is more stable than putting it in the ortho or the para position, which puts the positive charge directly adjacent to this carbonyl group. So while both of these slow down in reaction rate, the addition in the ortho and para positions, because of the instability of that positive charge, slows down even more. So we have a situation where now the meta position is more favorable than the ortho-para position because it's less affected by the positive charge in the intermediate. Ammonium type ions also tend to be very strongly deactivating. So if you think about aniline, remember aniline is aminobenzene. It has a nitrogen, aniline has 2 hydrogens on it like this, and then an aromatic ring. So aniline is a strongly activated ring, and aniline, the nitrogen, is an ortho-para director. But if you do this under acidic conditions, what ends up happening is the nitrogen gets protonated. The result is that this positive charge that we have on the nitrogen now tends to be very strongly electron withdrawing and destabilizes any positive charge that's put adjacent to it. So while aniline is an ortho-para directing, or the nitrogen is ortho-para directing in aniline, in an anilinium ion, that is one that has a positive charge, it's meta-directing. So where substituents get directed can change depending on the charge of the nitrogen on the ring. The last topic to cover is the effect of electron withdrawing and electron donating groups on the acidity of groups located on the benzene ring. So if you look at the diagram that I've given here, OCH3 is a strong electron donating group, and then subsequently these are lower electron donating. This is neutral because it's hydrogen. This is a electron withdrawing or deactivating, and this is a nitro group. If you look at the pKa's, you'll notice there's a progression from relatively high to relatively low pKa. So electron withdrawing groups in general decreases the pKa or increases the acid strength. So electron withdrawing groups increase the acid strength. The reason this happens is because when the proton is removed from the acidic functional group, there's a lone pair of electrons that is left behind, which can delocalize into the ring. But by having an electron withdrawing group, it turns out this creates a situation with additional resonance structures to stabilize the ion. Rather than going through the action of drawing all of them, I just wanted you to be familiar with this idea of electron withdrawing groups creating extra resonance structures. So you'll notice what happens here at this first structure. The oxygen's negative charge after the proton is released gets delocalized to this ortho carbon, and then it is going to delocalize to the para position, and then it can go on and go back up to the ortho position on the other side. That's this resonance structure here. But because this nitrogen is strongly electron withdrawing, by resonance you can delocalize one more pair of electrons to put the double bond here. So this is the extra resonance structure. This is the extra resonance structure that causes it to be more stable. Now let's try this. Let's see if you could rank these molecules in terms of pKa. We'll go from highest to lowest pKa. I'm going to pause the video and you look and you decide which one's going to be the lowest pKa and which one will be the highest pKa. Okay, so here's the answer. Phenol with no electron withdrawing groups will be the strongest or the weakest acid. So this is the highest. And then in the meta position, remember when you have a nitro group in the meta position or in the ortho position, it changes the position of the nitrogen relative to where the electrons end up in the resonance structures. So we have the extra resonance structure when we put it in the ortho position, but when we have it in the meta position, we lack the extra resonance structure. The result is meta position tends to be less acidic than ortho or para. The last topic to cover is how do we predict products or do the synthesis of disubstituted benzene rings? So I have two different products that I'd like to make shown on the slide. Both are disubstituted benzene rings. And what I need to do is do the retrosynthesis to determine what's the order in which I add the two substituents. Now, if you look at the substituents, for example, in the first product, I have a nitro group. The nitro is a meta-director, and the chlorine is ortho-para-directing. So how do I get the structure that's shown? And it turns out the answer is, well, since this is meta, that the meta group had to be on the ring before the ortho-para-directing group. In other words, retrosynthetically, I would go in this direction and I would say that this was the product immediately previous to the addition of the chloro group, the chlorine, and then prior to that I had benzene. So going in the forward direction, I nitrate the ring, that's HNO3 and H2SO4, and heat, and I end up putting a nitro group on the ring. And the nitro is a meta-directing, so when I add the iron, or the chlorine, Cl2, with FeCl3, I'll end up with the nitro group, and meta to the nitro group, I'll have a Cl. So let's look at the next product that we're considering. I have a nitro group and a chlorine group, but now they're ortho-para to each other, or para to each other. So what I need to do is when I do this reaction, I need to add the ortho-para director first, and then the nitro group will be added para to that. So again, retrosynthetically, just prior to this structure, I must have had a chlorine on the ring. And then I started from benzene. So working again in the forward direction, it's the same reactions. So I won't bother to draw the reagents again, but I'd add a chlorine, and then I would add a nitro group, because this is orthopara directing. I'd add a nitro group, and it would be the structure that I'm trying to build. Okay, so that's it for this chapter. There's actually quite a bit more we could talk about. There are some restrictions on the reactions that you choose to use, and there's many modifications of side chains or substituents that are added to benzene ring that we won't spend any time working on. But if you take the full class, the 28 A and B series, that's where you'll learn all the sort of nuances of synthesizing these benzene ring structures.